In this video, we learn more about the synapse. Neurons are able to communicate with each other through the use of chemical messages known as neurotransmitters. These neurotransmitters are passed from one neuron to another through structures called synapses. In the 1920s, Otto Lowery's experiments with frog hearts supported his hypothesis that the signal from nerve to muscle is delivered by a chemical. What he did was take a frog heart and a nerve that stimulates the heart, inducing it to beat slowly. On stimulating the vagus nerve that connects to the heart, he was able to reduce or slow the heartbeat after the stimulus. What he did next was collect the solution surrounding the nerve end and bathe a second frog heart in it. When he did that, he noticed that the heartbeat slowed down without the need of an external stimulus. This indicated to him that there were chemical messages present in the solution that might have caused the heartbeat to slow down. These chemical messengers were later called neurotransmitters. The connection or interface between two neurons is called the synapse. The first cell bordering on the synapse is called the presynaptic cell, and the second cell is called the postsynaptic cell. The presynaptic cell usually sends messages to the postsynaptic cell. These chemical messages, called neurotransmitters, are filled within vesicles in the presynaptic cell called synaptic vesicles. These are membranous sacs that contain neurotransmitter molecules. The region of the presynaptic cells that connects with the synapse is usually the axon terminal of the presynaptic cell. The part of the postsynaptic cell that connects with the synapse is usually a dendrite of the postsynaptic neuron. During signaling between neurons, an action potential comes down one neuron and then is passed through the synapse to the second neuron, which releases a set of neurotransmitter molecules that bind to receptors on the postsynaptic cell and lead to the generation of another action potential that travels through the postsynaptic cell and is passed on to the neighboring neuron. If we look more closely at the synapse, it's the place where signals from one neuron are passed to another neuron. The synapse connects the presynaptic neuron to the postsynaptic neuron. The gap between the two neurons is called the synaptic cleft, and neurotransmitter is contained in vesicles known as synaptic vesicles and is released into the synaptic cleft, which it crosses to reach receptors on the postsynaptic neuron. When an action potential arrives at the presynaptic terminal, local depolarization causes intake of extracellular calcium, which triggers the migration of presynaptic vesicles to the synaptic membrane. The action potential causes calcium ion channels to open and let calcium ions flood into the presynaptic cell, which triggers the fusion of synaptic vesicles with the presynaptic membrane, releasing neurotransmitter into the synaptic cleft. The binding of neurotransmitter with its receptor causes the ion channel to open. Inhibitory potentials allow intracellular potassium to flow out of the axon terminal, increasing the polarization within the postsynaptic terminal and making it less likely that an action potential will be triggered. This is one kind of potential that can be generated in the postsynaptic cell. The other kind of potential that can be generated is an excitatory postsynaptic potential, which allows extracellular sodium to flow into the axon terminal, causing local depolarization and bringing the axon closer to the threshold for the initiation of an action potential. Enzymes in the synaptic cleft break down the neurotransmitter once its work is done. And these degraded neurotransmitter molecules diffuse away or are transported into the presynaptic neuron via transporter proteins, where they are recycled to synthesize new neurotransmitter molecules. So, what do neurotransmitters do at the postsynaptic membrane? While a lot of neurotransmitters function as ligands, meaning that they bind their corresponding receptors called ligand-gated ion channels, leading to the opening of these channels and a flow of ions along an electrochemical gradient either inside or out of the postsynaptic cell. In this way, a chemical signal conducted by the neurotransmitter gets converted into an electrical signal, leading to a change in the postsynaptic membrane potential. Other neurotransmitters bind to receptors that activate enzymes for production of a second messenger. These second messengers are molecules produced inside the cell on the arrival of a chemical signal at the cell surface, and may trigger changes in gene expression, enzyme activity, or membrane potential. These changes in membrane potential can be one of two types, leading either to the depolarization or hyperpolarization of the membrane. Depolarization results from an influx of sodium ions, whereas hyperpolarization results from the efflux of potassium ions from the cell. Excitatory postsynaptic potentials, also known as EPSPs, cause the membrane to depolarize, increasing the likelihood of an action potential. On the other hand, inhibitory postsynaptic potentials, or IPSPs, cause the membrane to be hyperpolarized, decreasing the likelihood of an action potential. In this image, you can compare EPSPs to IPSPs and how they affect membrane potential. Note that an inflow of sodium ions is going to increase membrane potential, whereas an outflow of potassium ions or an inflow of chloride ions is going to hyperpolarize the membrane or make the membrane potential more negative. If these two occur at the same time, then they cancel each other out and the membrane potential does not change. Another thing to remember is that EPSPs and IPSPs are not all or none events like the action potential. These potentials are graded, meaning that they mildly stimulate the postsynaptic cell or mildly inhibit the postsynaptic cell, because this cell is receiving signals or messages from numerous neurons, and the sum of these signals is going to determine whether an action potential is generated in the postsynaptic cell. So these potentials, EPSPs and IPSPs, depend upon the amount of neurotransmitter released into the synaptic cleft and also the type of neurotransmitter released. Both of these potentials are short-lived because neurotransmitters bind only for a short period, that is reversibly, to channels in the postsynaptic cells. Since neurons typically make hundreds or thousands of synaptic connections with other cells, Lots and lots of EPSPs and IPSPs are coming into one particular neuron that is connected to all of these other neurons around it. And if these were to occur close together in space or time, the changes in membrane potential that result from EPSPs and IPSPs tend to cancel each other out. If several EPSPs occur close together, they sum up and make it more likely for the neuron to fire an action potential. This phenomenon is called summation. The sodium ion channels that trigger action potentials are located at the axon hillock within the postsynaptic cell. As we can see in this image, there are several axon terminals that are connecting to the dendrites of this postsynaptic cell, postsynaptic neuron, and also the cell body of this neuron. The signals they're sending are either excitatory or inhibitory. The effect of these potentials on this postsynaptic neuron is going to be the sum total of all of these potentials. If the membrane of the postsynaptic cell depolarizes past the threshold potential, an action potential will begin and will be propagated down the axon to the next synapse. So if you look at a graph of the change in membrane potential as these EPSPs and IPSPs come in, the excitatory potentials are going to add up if they occur close together in time, but these still don't reach the threshold potential, which is around minus 55 millivolts. And if these EPSPs occur close in time and bring the membrane potential closer to the threshold, makes it more likely for an action potential to result in the postsynaptic cell. This brings us to the end of this video on the synapse. After going through this information, I'd like you to be able to answer the questions listed on this slide. Thank you.